Unsustainable human activities and natural disturbances have resulted in the degradation of nearly 70% of the world's ecosystems. Ecological restoration has emerged as a key tool for combating biodiversity loss and reestablishing ecological function to damaged ecosystems. However, the establishment of native plants and restoration projects may be hindered by a variety of stressors, including drought and invasive species. As both drought and invasive species pressure are expected to increase in coming decades, understanding how they interact is imperative for identifying barriers to successful revegetation. One particularly problematic invasive in the western U.S. is the annual grass cheatgrass. It begins growing very early in the season and may use up resources such as soil moisture to the detriment of establishing native plants. We worked at 2 sites in Colorado, one on the eastern slope and one on the western slope of the state, to assess the independent and combined effects of growing season drought and cheatgrass on native plant establishment and soil moisture. We found that drought decreased cheatgrass abundance at the western slope site, but increased cheatgrass abundance at the eastern slope site. Both drought and cheatgrass reduced native species abundance, but in some cases effects depended on whether native plants were forbs or grasses. Both drought and cheatgrass reduced soil moisture, but effects varied considerably by year and by site. We were particularly surprised by 2 findings. First, at the eastern slope site, we found that drought resulted in lower soil moisture in the summer, but higher soil moisture in the spring. Second, at the western slope site, we found that cheatgrass reduced soil moisture to a greater extent than drought in the first year of our study. This research demonstrates that both drought and invasive species can negatively influence native plant establishment and soil moisture in restoration settings, and in some cases may interact to do so. But because of the variable results between our 2 study sites, additional research is needed to better untangle these interactions and their effects on revegetation outcomes.